So recently I went on In the Wheat of the Conversation with Jackson Wheat and Dapper Dinosaur, and I think the conversation went great. I had a really great time on there, and I'm really happy that they invited me on. I'm honored to participate in the discussion, and I'm happy to come on in a second time. Now, I think there were a few arguments and a few claims made in that video that need to be addressed, and a few topics that we didn't get to that I think we should elaborate on further. So I'd like to do that in this video. So if you'll join me, I'd like to go through a couple of these claims and a couple of arguments that were made/presented on both sides that I think we can really go through a little bit more in detail and understand. Well, let's get started.
We ask your help in this fateful moment as sign that we scientists do not stand alone.
I agree.
We passed the 6-minute mark in our countdown for Apollo 11. The flight to land of the first men on the moon. We're on time at the present.
And I put in it one living cell.
Over time, you will come closer together, ultimately bumping into each other at the North Pole.
So journey with me to the sun.
Of sandstone, shale, limestone that goes continent wide, there are 4 other big sequence packages of strata.
Let us go faster. Bear with me.
It's as though there was a force pushing you together.
DNA contains a similar number of genes that directly code for proteins as a lot of simpler life forms do.
No one has measured the speed of light. That's ridiculous.
World and other continents in that general style.
Faster. Faster.
The real reason for you coming together was that you were both on straight paths, geodesics, on a curved surface.
What is time?
A well-documented find of modern biology is that DNA is the language of life. That inside of every single one of your cells is an intricate and complex coding system for your entire existence. And it has come to my attention that not everyone acknowledges or understands this concept. And it seems to come from the confusion of how to define complexity and information in biological systems. So, how do we define information? Well, there are 2 ways. So the first of the 2 ways is the method known as digitization. This is when you code information into a binary digit system in order to count how many bits of information are contained within a given string of letters. This is the same way that computers communicate, turning instructions or language into ones and zeros, which then quantifies the information for the computer to read and count. This very same concept can be applied to the 4-letter language of DNA. The second method involves calculating the amount of entropy or randomness or disorder in any given string of information. In order to achieve this, we use the standard informational theory model, also known as the Shannon informational theory model. With these 2 measures, we can quantify the amount of information and complexity in an organism's genome, and we have. When the entropies of species genomes are compared, we can measure their relative complexities and often find that marsupials, primates, and other mammals are the most complex. So when I hear someone say...
That's another thing that I don't really know how to measure. I don't know how to compare complexity in a human versus a tomato plant.
It's not because we cannot do it. Just for example, like tomato plants, I believe, have more genes than humans do.
Yes, by big margin.
And I don't think any competent biologist would tell you that a tomato plant is more complex than a human body. And so...
That's another thing that I don't really know how to measure. I don't know how to compare complexity in a human versus a tomato plant.
Have you ever used a mousetrap? I promise this is related. Just follow along with me. If you have ever used a contraption like a mousetrap, you may have noticed that a mousetrap has many unique parts that over time, these parts eventually wear away or break, leading to the non-functional mousetrap, as in it cannot perform its normal function. This is due to the fact that in order to fulfill its function, all parts must be present and working together. This property of not being able to remove or damage a part of a system without maintaining the function of the system, in this case, the mousetrap, means that the mousetrap cannot simply be built one piece at a time. When we talk about biological systems, many biologists refer to this property as being called irreducibly complex. Now, it was stated in this discussion that
such systems have evolved under direct human observation within historical timescales.
Yet, it was also claimed that...
And I actually do think that many of these systems are irreducibly complex under the definition B he gave. Like, I would not argue that the blood clotting cascade or the flagellar modem...
You have to have all the parts for it to work. Right, exactly.
So what's going on here? Let's address each point, yeah? First, the first claim that we have observed these systems evolved is incorrect for two reasons. One, we have not in fact observed irreducibly complex systems evolve, let alone observed non-irreducibly complex systems creating irreducibly complex systems from scratch, as in an irreducibly complex system coming from like dead matter. Let's just use that as an example. And two, the arguments often used against this fact often include A, instead of gaining a part via natural selection, the organism/system lost a part, thus leaving a system that is irreducibly complex. And B, the system was originally used for a different function and gained a part to fulfill a new function. Can you see a problem with either of these two arguments? You see, they just push the problem back a step, as someone can then ask either A, why/how does the system gain an extra part in the first place to lose? And on top of that, you still haven't accounted for the set of parts that are still required to be there for the function to be fulfilled. And B, again, how do you account for the original system being used for a different function? You have also still failed to account for the generation of the new part in order for the system to fulfill the new function. As in, when a previously irreducibly complex system becomes a new irreducibly complex system and they have different functions, there is still an added function that needs to be that is required in order for that new irreducibly complex system to fulfill its function. Now, critics of irreducibly complexity will also make the claim that
subfunctionalization of any gene basically is irreducible complex, or results in irreducible complexity.
Yeah.
Which supposedly at least accounts for the generation of a new function once we have an original irreducibly complex system, and to add the function to. However, this claim is also incorrect. Not only do the majority of subfunctionalizations do nothing or are deleterious, all subfunctionalizations that acquire a new function also maintain the original function of the gene. This means that, for example, if the bacterial flagellum originated from another molecular machine, the original would retain much of its original function while also gaining an entirely new function. This is not what is observed. The bacterial flagellum, as far as we can tell, only has its primary function as the locomotive/sensory organelle we know it for. So now that we've reviewed a few of the points, I'd like to address the biggest topic that we didn't get to, which is abiogenesis. This is a point that...
So a couple of things. First, typically what Dapper and I do when we have these conversations is we usually skip abiogenesis, because we usually just talk about, like I said earlier.
And I don't blame them. The reasons why? Let me tell you. Because mathematicians have calculated the probabilities of events such as the arrangement of the first functional amino acid chain, and have found it to be virtually impossible given the accepted age of the universe. To give you a sense of the probabilities involved, the informational theorist Hubert Yockey, who worked under Oppenheimer on the Manhattan Project, calculated the probability of just a small protein called cytochrome C, which according to convention had to appear very early on in the evolutionary history of cells. Yet, he found that even in an amino acid-rich environment, the probability of a simple cytochrome C arranging itself spontaneously is around 10 raised to the negative 75th power. For perspective, this is less likely than winning the Powerball lottery 9 weeks in a row, only putting in 1 ticket per week. And that's for one small protein. For one of your cells requires thousands of proteins just to survive. So the probabilities quickly stack up. Well, I'm sure most of you will have lots of questions about this topic and the stuff that I've discussed in this video. And I know it wasn't exactly the longest video, but I'm working on other videos at the moment right now. Stay tuned for Monday Mythbusting. The topic's going to be Flat Earth, so that should be entertaining. But if you have any questions or comments on this video, feel free to leave them below. I'm going to link all the sources in the description, and I will catch you on the next episode of Science Time with Terry.